All right, so today for crop science, we're going to talk about seed germination. So looking at this, we're going to identify parts of a seed, describe their functions, the process of germination, the conditions required, and the importance of seed quality. So parts of a seed, there's three primary parts. You have the embryo, endosperma, and seed coat, and you'll see pictures of these in upcoming slides. So the primary parts, you have embryo, basically it's the young plant before it starts to grow, before it starts to emerge from that seed itself. Basically, any seed packet, those seeds sitting in a cabinet or wherever they are, hopefully in a dry, cool location, it's got all the genetic information it needs. It just needs the right conditions to start growing. Part of that seed, so the second part, you have embryo for the first one. Second one is endosperm. That's the source of food. It's going to help nourish that plant as it grows. That's why you can start seeds in water and they'll start to grow. But eventually it's going to need nutrition from somewhere else through the roots. Then the seed coat, there's one basically protective layer, we might be one or more, that are around the seed. And some seeds as they grow, you'll see it pop off of them. Some of them, it might be really thin and get damaged easily in shipping. Okay, so seeds begin to form in embryo after fertilization. So after that pollination happens, and it does say it starts the zygote. The initial division of the zygote results in two cells. The bottom cell develops into a multicellular structure called the suspensor. The suspensor anchors the embryo and helps to obtain nutrients from the endosperm. The top cell develops into the embryo. So the first cell divisions from this top cell create a chain of cells called the proembryo. As the cell divisions continue, a globular embryo takes shape. At this point of development, the cells begin to differentiate. So they start to change. Cotyledons begin to form. So that's the first true leaves that come out. So think of those monocots and dicots. That really long word is typically monocotyledon or dicotyledon. They're just using the general term of cotyledon. It's a seed leaf that stores food in the form of starch and protein for use in the embryo. Embryos of the monocotyledon, so monocot plants, have one. Dicotyledons or dicots have two true leaves. Monocot plants store the bulk of their energy in the endosperm. Dicots store their food in the two cotyledons. The cotyledons grow and elongate as the embryo matures. The pressure of the expanding embryo crushes the suspensor. So you can see there's your comparison of dicots and monocots. They're just kind of showing you the difference. And we've talked about before how the dicots tend to split apart easily. The monocots have that gigantic endosperm to serve as that food source too. Skipped one. This is just showing you kind of a zoomed in example. So you have back to that flowering piece of the plant. They're showing you where the zygote forms would be in the ovary. So this is that female part called a pistil. You have stigma style and then ovary, and that's where the seeds start to form. Depending on the plant, they look very different. There might be multiple in a pod. Which actually grew from a flower, so they all look very different. So it's just showing you to give you more visual aspect as to how that cell division takes place. Okay, so the parts, still going through those parts, the mature embryo consists of an embryonic root known as the radicle, an embryonic shoot, and one or two cotyledons. The embryonic shoot, known as a plumule, has two main parts, the epicotyl and the hypocotyl. The epicotyl is the portion of the embryonic stem above the point at which the stem is attached to the cotyledons. The hypocotyl is the portion below the point of attachment and is connected to the radicle. Seed encased and protective coat, that's kind of nature's way of protecting it. So if it gets shipped or carried by plants, animals, it's going to be protected and not open right away or be damaged by, maybe extreme temperatures. It does talk about here, it says the scar or a scar called the helium can be seen at the end or along the side of the seed coat. The helium marks the point of attachment of the seed to the ovary wall. To me, the helium, I think of it as a belly button. Some seeds, it's really evident. So if you think of like black-eyed peas, it has a big dark spot. Soybeans, it's kind of the reverse where it's a tan color with a dark spot. That's actually where the helium is. The seed coat has a tiny opening, sometimes visible near the helium, called the micropyle. So sometimes you can see it almost looks like a seam on the seed is what it's getting at. So the next part, we've gone through the parts. This part is all about germination. So germination is, think of the plant growing. So what conditions does it need? If you look at the picture on the screen, notice they're adding water to it. Oftentimes we associate water with plant growth. The hard part for many of us is to think about the fact that water does not equal love. You can water plants way too much and basically drown them. You might cause them to rot and mold. So keep in mind they have to have the right conditions. And then obviously it says many important crops are grown from seeds. Regardless of your background, you've probably driven by fields and noticed that there's soybeans or corn or wheat. They're all started from seeds. It's much easier to ship them that way, and it works quite well. Some plants produce seeds which germinate immediately once they're released. Others produce seeds that have internal dormancy mechanisms and remain dormant until conditions are favorable for growth. So it's really interesting to me if you think of something like an oak tree that drops acorns. They actually need a freezing portion to actually go through the process of germination. So the tree will drop the seeds. Oftentimes animals, stash them, bury them, and then it still has to go through that cold snap. Once the cold snap is over, that seedling will start to grow. So it's, kind of to think about what nature does to some of these plants naturally and how do still make it. So this is stratification. It's a dormancy mechanism that involves temperature. Seeds with this mechanism must experience a period of cold. So just like we talked about the oak tree, they have to experience cold before they can germinate. So remember, germinate is just starting to grow. Scarification is a dormancy mechanism that involves the breakdown of the seed coat. Some plants have a very thick, tough, protective seed coat. So we don't think about this, but even like an avocado, the seed is, past that fleshy meat of the plant. It could be like a peach. The pit is in the middle. Those are actually big seed coats if you think about it that way. And some of them even have an inner layer within that seed itself. And honestly, it does say too, these seed coats prevent water and oxygen from entering the seed. Another thing you might think of is all of the tree nuts, like the walnuts, things like that are really thick. And then the seed coat must be broken before germination can begin. Major stages in germination. So germination begins with the seed's absorption of water. Most dormant seeds have a 5 to 10% moisture content. Those of you that are in the grain world or, have family that grain farm know that moisture can sometimes be your enemy for storage. So that's why it's at most have 5 to 10%. You wouldn't even really notice it. As the cells hydrate, they swell and become what they call turgid or rigid. The moisture triggers an increase in the cellular respiration. In stage 2, metabolic activity surges, proteins are synthesized, and this is where you should see a spurt of growth. The sugars and amino acids are directed towards cell division, growth, and differentiation sites at the root and shoot tips. Metabolic processes increase in the third phase of germination. The swelling of cells causes the seed coat to rupture. The radicle emerges downward, so it's the start of the root system, and then the stem goes upward. Remember, this is that whole idea of if I plant my seed too deep, it's not going to grow because it can't, start that process of photosynthesis. It can't figure out where up is. The shoot begins manufacturing food through photosynthesis. The roots absorb water and nutrients. So this is showing you that germination process in kind of a chart form. Goes through all of those steps and how it gets to basically go from seed all the way to the plant. All right, so germination begins when favorable conditions exist for the survival of the developing plant. Conditions of germination include moisture, air, optimal temperatures, and possibly light or darkness. It really does depend on the plant. The water triggers germination process and is necessary as the embryo grows and develops. So you can kind of see they're showing you a plant cell, and as that vacuole fills with water, obviously it's going to start to change shape. All right, so temperatures, it can be anywhere from 32 degrees to 105 degrees. Optimal temperature for the majority of our plants is 65 to 80. Temperature influences the speed of metabolic activities. Metabolism's faster when temperatures are warm than when temperatures are cool. So even right now, if you have some houseplants, they've kind of slowed down because it's wintertime, it's cool out, they are not as active when it's cooler. And this is showing you enzyme activity during germination, all the different things that happen throughout. So it says things like amylase, that's basically sugar. Gibberellins is actually a growth hormone. So there's all these different things that naturally occur within a plant. All right, seed quality. So if you know anybody that does grain farming, they tend to buy seeds from well-known companies that can back their product. If you have someone that has a vegetable garden, they typically buy from a company. And honestly, if they're like me, I save seed packets so I know where I bought from the previous year and which varieties I bought. So I know that, yep, it went well. Honestly, if I have something that doesn't germinate well, I tend to throw the packet so I don't buy that again. So there's something to think about. And notice it does say seed quality refers to both viability and vigor. So viability is the ability of seeds to germinate under optimal conditions. Vigor is basically that plant being able to germinate under different conditions. So if the conditions aren't quite ideal, can they still produce a healthy plant? So companies, if you look at any seed packet, and you guys are going to be making one today, they test their seeds to determine the percentage. So they can determine the percentage that will actually grow. Obviously, the higher the percentage, the better off the farmer would be, or, gardener. And then notice it talks about storage. So the goal to maintain seed viability and vigor during storage period, the best seed storage conditions typically consist of cool temperatures, about 40 degrees Fahrenheit, and low humidity, approximately 15%. All right. So these are some questions that if we were in person, we'd obviously go through, but things that might come up again on a quiz. So parts of the seed germination, what is it? We will eventually do a germination test so you guys can see what it looks like when plants start to sprout, because it's something that many of us haven't experienced. And then what conditions are required for seed germination? And then why is seed quality important? So that's kind of just wrapping up the whole idea of seeds starting to grow. You're going to go through and make a seed packet, and we'll actually, start to hit on things like nutrients. We'll look at even the importance of temperature and how that affects plants, and you'll see it when we go through the different crops as well. So thank you for listening today, and tune in next time.